Welcome to the second episode of Dalia Science Neuro Shorts. We're going to be discussing some further aspects and elements of the nervous system today. We will be looking at the continuation from what we've learned before. Hello, Filosofnik, good to see you watching. That's very nice of you. I'm seeing some more people joining. Very happy to see that. I'll try to remind you about some stuff that we discussed last time, and then we're going to see some more things about the fly nervous system. Actually, no, we're not going to be talking so much about the fly nervous system today. We're going to be talking about the development of the nervous system in general, and we're going to be looking at what happens when the organism first has the chance to develop its nervous system and how it looks. If we were to look at some graphs and if we were to look at some schemes, which will help us understand how the development of the nervous system happens. So last time you might remember that we talked about why people use flies in research in the first place, because they are amazing model organisms and you can look at all kinds of developmental defects, all kinds of diseases using flies. We were also talking about what the nervous system is and what it's composed of. And we were also talking about why flies are particularly suited to look at the nervous system of all the model organisms we can have. And so today we're going to be sort of continuing on that, as I said the last time. First of all, these are not exactly like Oxford tutorials. They are just talks from me, an Oxford tutor, telling you a little bit about what people learn in tutorials and so on. And second of all, I'll probably be able to see some of the questions as we go, but I'll be pausing from time to time and scrolling up and down trying to see people's questions. I hope that's fine. So anyway, welcome to episode 2. We're going to look at the nervous system development today. It's really exciting. So as any system in any body, in any organism, in anything, the nervous system has to develop. So it has to be formed from what simply was an embryo, from what was a zygote, until it's fully formed, until we've got a brain, until we've got other elements of the nervous system. And there are complicated processes which are involved in this whole situation and how it happens. And so we're going to sort of walk through them and try to understand them a little bit better today. So first of all, an interesting fact: the human brain has 10 to the power of 11 brains. The human brain has 10 to the power of 11 neurons. That's a lot of neurons. And those neurons, you can see I wrote out the number of neurons that there is. It's like a lot. Actually, I'm gonna get a pointer. 1 second. There's a pointer. Here we go. Yeah. So as you see, that's like a ton of neurons. That's a lot of cells. And those cells obviously have to talk to each other. So the whole point of a nervous system is to send signals one way or another, to talk to each other, to let the cells communicate. So for example, even when I'm moving my hand right now, my nervous system is in full swing doing things, letting my muscles move, letting me pick up my pointer and letting me talk to you. So obviously, the whole point of the nervous system and of the neurons is to be connected to each other. And interestingly, each neuron makes around 1,000 connections, which I think is quite a lot. So every single one of those 10 to 11 neurons in the brain makes 1,000 connections. So that means that is 10 to the power of 11 plus 10 to the times 10 to the power of 3. That is 10 to the power of 33 connections in the brain. That is insane. That's 1 and 33 zeros, connections in our brain. Those connections obviously are synapses. We talked a little bit about that the last time. And here is an image of a real neuron. And so that's interesting because whenever I look at this picture, by the way, the pictures that we're looking at today are from Albert's Molecular and Cell Biology, a very good textbook, so I can share what this textbook is with you later on the stories. So anyway, so this is an image sort of a real neuron. It's like a projection taken from microscopy images, which is shown here. And I always find it incredible to look at it because basically this neuron here shows how many different connections one neuron shows. And then, like, if we looked at like a cartoon representation of a neuron, like we looked at one last time, well, it kind of simplifies how complicated the neural network is. And here we can see that even on the side of it, there are multiple connections being created between this neuron and other neurons, and not only at the nearby the cell body and nearby the ends of the neurons. So I think that's like a much more realistic presentation of what a neuron looks like this, and basically of how neurons behave in the real working nervous system. So I think it's much easier to understand from this real representation of how complex really the nervous system is. So anyway, the development of the nervous system happens in 4 main phases. And we will discuss those phases because it's important to understand that it's not like a process which happens overnight. It's a thing that's gradual. And a lot of important molecular signaling and a lot of important processes have to happen for this system to form. So the first phase of the development of the nervous system, we could imagine it is the neurons are born. So obviously, the neurons have to come from somewhere. And they come from cells, which we call the neural stem cells. So those neurons are born and they are called the neuroprogenitors. Progenitor is a kind of a cell that isn't a fully formed proper cell yet. So it's not a neuron yet, but it's got the potential to become the neuron. And here we can see there's something else that's represented, and that's the target cells. So we've got the neuroprogenitors and we've got the target cells. And those future neurons are born. They are ready to start doing what they are best at, which is make connections. So we've got those neuroprogenitors, we've got the target cells, and the first step of the neural nervous system development is the birth of the neural progenitors, of the future neurons. And then the second phase is that these neurons will extend their axons and their dendrites. And we saw the last time where the axons are and where the dendrites are in the neurons. And if we came back to this slide, so obviously we've got the dendrites surrounding here, the cell body, the cell soma. These are also dendrites. But then this longest projection here that we're looking at, this is the axon. And here we've got the synapse. So those neurons will extend their axons and their dendrites to try to make their way towards those target cells that they want to connect with. So that's the next thing that's going to happen. So that's phase 2 of our nervous system development. These future neurons start to grow towards their targets. And here we've got a representation of that. We can see that there are those axons which are kind of extending. You can see the red arrow here, and they're extending and making their way because they're trying to find out where their targets are. And this extension, this process of trying to find their targets, can be guided by different things. It can be guided by chemical cues, it can be guided by mechanical cues, it can be guided by both. What the cues mean, they're just sort of like tips, like signposts for the axons to find their way along that road. So if you're driving on any street in Dukoy or wherever, you can see some signs which say, for example, speed limit, or you can see signs that tell you, oh, like turn here for London. And this is basically the sort of a cue that the nervous system, the cells in the nervous system get from their targets to make their way towards the target. So they can sense some chemicals that the targets are producing, and then they can also sense some cues, mechanical things. So for example, oh, the road is really rough here, or for example, they can see that there's a little bump. And so those mechanical cues are also something that the neurons can sense. And in that way, they are able to find their targets in the nervous system. And finally, once they found those targets, thanks to those chemical and those mechanical cues, there are synapses that are formed. And we looked at a synapse last time. A synapse is basically a connection between a neuron and a neuron, or a neuron and some kind of an effector like a gland or a muscle. And in a synapse, information is being conveyed from that neuron that forms that synapse and with the other neuron. And it's being conveyed from the first originating neuron to the second one, or from the neuron to the muscle. So this is the synapse. This is like a connection between two neurons. And so after neurons are born, after they found their way to their targets, they will connect to the target via a synapse in order to be able to exchange information with their targets. So this is the third phase of this nervous system development, basically. And we can see that sort of here. So here we can see that some of these synapses, some of these neurons already formed like the little green endings, and then these are the synapses obviously, and some of them are still growing towards their targets and trying to find their way. So this is phase 3. This is where the neurons use different guidance cues to find their targets and then to connect with it using synapses. And then finally, there is this element which is just the synapse remodeling, and this is the phase 4 of the development. And it's really interesting because the phases 1, 2 and 3, which we sort of discussed in the beginning, they happen during early development, during fetal development, and when the organism is very young. But then this fourth phase, the synapse remodeling, happens throughout the entire life. And that's very interesting because this is what I work on. So an element of that synapse remodeling and refining is synaptic plasticity. I'm seeing a question here from Osama Anbarji, sorry if I'm saying it wrong. The others will die if it doesn't have a stimulus. Yes, that's true. So for example, one of the things that happens with those neurons, we can go back to this image. So some of these, they will never find their target. They will try to go and find the target, but the chemical cues and the mechanical cues maybe won't be good enough for it, or maybe it just gets lost. And actually it's been found that the nervous system overshoots the number of neurons that are needed to connect to some target organ. So it just produces more of these neurons than are necessary, and some of them will connect and the rest of them will atrophy and die. And so that's an element also of that remodeling of the synapses. So some synapses are made and then it turns out that they are not necessary, while it might also turn out that a synapse is needed where there isn't one. And so, for example, we could think about it on an example of learning and memory. If I learn about something today, so for example, one of you might learn about something about neurons, but if you don't repeat it and if you don't remember it and if you don't read more about it, that connection in your brain is going to be severed. It's going to disappear. So that's an example of synapse remodeling. So forgetting is synapse remodeling. I've got a question here. Can one neuron connect to more than one target, I'm assuming? From Sauhard Rishi. Sorry if I'm saying it wrong. Yes, we were saying in the beginning that every single neuron in the human brain, so there's 10 to 11 neurons, and each one of them makes around 1,000 connections to different neurons. Actually, there is this behavior in neurons as well, which is called self-avoidance. And that behavior dictates that the neuron doesn't make connections to itself, because neuron making connections to itself is quite pointless because the network cannot be created in the nervous system. What chemicals are released to attract the neuron to a target cell? An example are neurotrophins. They are growth factors, which are one of the very first discovered growth factors in the nervous system, which are released by the target organs, and they promote the growth and the extension of the neurons, basically. And they are used in a lot of experiments, even in the lab, to keep the neurons alive and happy and keep them growing. Will you be able to design a synapses model ex vivo for ALS disease? There are people who work, this is a question from Ozan Fidan. There are people who work on ALS, on cell models, but usually you would use some kind of an animal model because it's quite a complicated disease. And basically ALS, so that's a type of a motor neuron disease. So it's a disease in which a motor neuron is not properly connected and the motor neuron doesn't function correctly on the neuromuscular junction, which, as you might remember, is the connection between the neuron and the muscle. And so, for example, a lot of people work on fruit flies on ALS because it's just so much easier. It's extremely hard and it would be not efficient and not as good of a model to try to look at that using a cell model. But yeah, people do use cells in ALS research. Yeah, and it's synaptic pruning. Basically, that's what I said. So Alarbilges, sorry, I don't know how to pronounce your username, guys, is saying that it's synaptic pruning. And yeah, basically this is synaptic pruning. So synaptic pruning is really important in both development, but also in synaptic remodeling in an adult life. Can one neuron connect to a particular target cell with more than one connection? Yes, it can. It will depend on the function of the organ that it's connecting to and on the structure of the neural network at that point. So this whole discussion that we just had falls into the category of the phase 4, which is the synaptic remodeling and synaptic plasticity. And this is the final phase of the development of the nervous system. But because it lasts through adulthood, we could say that the nervous system develops from the day we're born until the day we die. Because the synapses always get remodeled, and when we learn a new thing, that's a synaptic plasticity example. And when we forget something, that's also synaptic plasticity. So it's really interesting. Wonderful. Let me see. Yes. Oh, and I think we had a slide here. Yes, basically this slide sort of shows that these neurons are already connected to their targets and they've got this network going on and it's like being created, and some of those can get removed by synaptic pruning and synaptic plasticity. Wonderful. So we're going to move to the development of the central nervous system, and we're going to discuss it on an example of one of the elements of the central nervous system because we simply don't have enough time to just talk about it forever. Regardless, central nervous system originates in the neural tube. What's the neural tube? That's what it is. This is neural tube. So it's this structure which basically develops at a very early stage of development, and it is made of the very early epithelium that exists in the developing organism. And we will have an example of that in a minute, but the entire spinal cord, the eyes, the retina, the brain, originate at that neural tube. So that's very interesting to see because it's this very simple structure, and then towards the end of the development it forms those complicated organs like a brain. There's another question from Sahar Rishi. How do synapses regrow after being destroyed due to some accident? They don't, and that's one of the most problematic things in science and research. As in, it's wrong to say they don't. Sometimes they do, but in most cases they don't. This is why, for example, people who are paralyzed, they don't stop being paralyzed most of the time without therapy, because it's extremely hard to re-establish a very complicated structure present, for example, in the spinal cord or in the hand, and reconnect the nerve. And so actually one of the reviews that I wrote in my undergraduate degree was about neural engineering. And this is when people do research and try to develop implants which could fill in a gap in a nerve that, for example, has been broken, because there is something that's called critical gap. So if there is a break in the nerve, in the neurons, due to the accident, if it's small enough, it can repair. But if it's bigger than 1 centimeter in humans and in rats and in mice, it won't repair on its own. So there are people who use clever materials, for example, those that conduct electricity, or materials which contain neurotrophins, as we mentioned earlier, to try to get neurons to grow from one end of that damaged nerve to the other and fill in that gap. So yeah, they sort of don't, but they can if they're aided with research. We're back to the neural tube. This is the primary structure from which the brain, the spinal cord, and the retina develop. And this is a scheme which kind of shows how it happens at a very early stage of development. So we've got this sheath of epithelial cells, which form very rarely after an embryo is formed. And basically, the first stage of formation of that neural tube is this invagination of epithelial cells, and is caused by the tightening along those ends here, which are the adhesion belts. And then this invagination kind of caves in, and the epithelial tube then like pinches off, kind of separates from that layer of epithelial cells, and there's this like tube in the end, and that is our neural tube. So this is how that process occurs at a very early stage of development. And we get the resulting structure, the neural tube, undergoes further changes, which we will discuss in a second, to be able to form the structures of the nervous system. Someone's saying, guys can look up the difference between, it's Zach Dalton, between oligodendrocytes and Schwann cells. It contributes one of the reasons why CNS and PNS have different regeneration capacities. Yeah, that's very true. Oligodendrocytes are glial cells of the central nervous system, and Schwann cells are the glial cells of the peripheral nervous system. And there are differences in how they behave upon damage. When damage is happening in the peripheral nervous system, Schwann cells detach from the axons to which they were attached before, and they can clear up debris and also start differentiating in order to become neurons and fill in the gap. But oligodendrocytes have less of that capacity, so that's one of the big differences. And that's actually what my main project is about, because I work on glia, on the flies. Anyway, so we've got the neural tube, which is very exciting, but we don't really care about the neural tube. It's not exactly an exciting structure from our point of view as a neuroscientist. We would like to find out how this neural tube becomes something else. So for example, cerebral cortex. First of all, what's cerebral cortex? It's the outer layer of the neural tissue of the cerebrum in human mammal brain. So human and mammal brains have the structure, and it's called the cerebral cortex. And this definition of cerebral cortex is explained using another word which we might not know. I don't know who's watching me, so I'm not going to assume we know it, but the word is cerebrum. And cerebrum is just basically this uppermost, largest part of the brain, And we've got that presented here. It's like the stereotypical image of a brain when you learn about it in school or at uni. This is the cerebrum. And the cerebral cortex is actually the white matter of the cerebrum. So white matter usually is the glial cells and the axons, and the grey matter usually is the cell bodies. And so cerebral cortex is the axons, the sort of projections of the neurons in the cerebrum. So this is where we're at. And this the cerebral cortex, the cerebrum has all these elements, the lobes and so on in here, which we could look at in more detail, but not today, because today we're looking at development. So we're looking at how this develops, how this cerebral cortex develops, because the cerebral cortex is really important in terms of how intelligent humans are and how intelligent other animals are. This is where most of the cognition happens. So the cerebral cortex originates in the anterior neural tube. So anterior and posterior just refers to the body organization of any sort of organism. And anterior just means more close to the front. So anterior for me, for my person, this is the anterior end of my person. And then the posterior end of me would be my feet. So anterior is close to the front, posterior is close to the back. We also have this differentiation, which is important to remember, and that is dorsal and ventral. So dorsal for me is front, close to my belly, and ventral, wrong. Dorsal is close to my back, where my spine is, and ventral is where my belly is. So this is how you refer to any organism developing when you want to describe geographically where something is on its body plan. So here we've got the statement, which is the cerebral cortex originates in the anterior neural tube. So that means that the cerebral cortex, which is an element of the brain, originates towards the front of the neural tube, towards the front of the body of the organism in the neural tube, which is perfectly, I would say, logical, because the brain is towards the front of any organism, like myself. So we're going to move on. This is the first element, this is the first stage of the development of the cerebral cortex. The anterior neural tube cells keep dividing, divide and divide and divide. This neuroepithelium, so epithelium is the tissue from which the neural tube originates, and now it becomes the neuroepithelium, so it's like it's tissue which is connected to the future nervous system. So this neuroepithelium thickens, becomes bigger, and expands as the cells divide. So let's move on. This is kind of a graph that explains this, but I'm going to see, because I've got a question here. It's a question about the eye that I'm not an expert on. When there is light in the cones and it doesn't release glutamate, that makes the next neuron work. This is the question, how the brain knows that their stimulation is something to see. Sorry about that, I really don't understand this question, Osama, because it's about the eye and I'm not an expert on the eye, really. So maybe you can private message me after the live and we can try to chat about that if that's better. Coming back to the nervous system development and the neural tube. We've seen this sentence which we looked at before, which is that the anterior neural tube cells divide, and this is the process in which the cortex is going to be formed. And here we've got a representation of this neural tube, and if we wanted to, so you remember that kind of little graph where it was like a little circle, where this is basically what it is. And we can see that this embryonic cerebral cortex is kind of that element of that neural tube. And then the future neurons in that little neural tube, they're not really neurons yet, but basically the future neurons are born close to the lumen. And now what is the lumen? The lumen is that opening, that kind of gap in the neural tube. It's that like space, the empty space. So the future neurons are born close here to the neural tube lumen, and then they migrate outwards. Thank you, April, for telling me that you like my talk. And anyway, so these future neurons are born close to the lumen, which is this opening, and they migrate outwards. So they go from the inside of that tube towards the outside. So here we can see that process basically, and I don't know how well you can see it, but I can try to kind of get my phone and make it better. Yes, there we go. So we've got that neural tube, and the cells are born here and they migrate outwards. Let me try to focus it. Is it focusing? Yes. And then this is a representation of it. So basically what we've got here is there are those kind of future neurons, and they're like those red kind of dots. They look a bit like sperm cells, which I think is kind of funny. Anyway, and then they migrate outwards. So if we compared it to this graph, they migrate from the middle of the neural tube towards the outside of the neural tube. So this is what it looks like. Okay, let me try to re-establish my phone. Oops. There we go. Is that fine? Yes. Okay. So we've got those neurons, and they migrate from the inside of the neural tube towards the outside of the neural tube. And then the third stage of the development of the cerebral cortex is that those cells which just now migrated, they form radial glia. And despite the fact that they're called glia, they're not glial cells. They're not glial tissue. They are progenitor cells, which means that they are stem cells which will develop into something else later. And those radial glia are the neuron and glia precursors. So they are a form of a neural stem cell. So in the further stages of development, they will develop neurons and into glia. So this is our third stage of the cortex development. And then here we can kind of see that. I'll try to grab my phone again. So we saw those migrating cells here, and they were migrating upwards and outside the neural tube. And then finally here, there's like a little graph which kind of shows this. So there's the nucleus of those radial glia. This is a migrating kind of neuron, which is that precursor. And then this is the radial glial cell process. So worth mentioning is the fact that most, if not all, cells of the nervous system form something that's called processes. And those processes basically are like little feet, little extensions. And so here we can see a radial glial process. And thanks to those processes, those cells crawl, they change their spatial location, they change different things. Let me fix there we go. Okay, so this is our third stage of the cerebral cortex development. And then finally, there's a stage at which we kind of see the bigger picture, and that is the successive generations of neurons build up a series of cortical layers. So their cortex will be basically made of layers, and we can see it here on this little graph. So basically, and I'm going to grab my phone now. I apologize. There we go. So here we can see that in the beginning, we've got those dividing progenitor cells, and they will migrate upwards and form neurons in layer 1. So this is layer 1. So they divide, crawl up, and they form layer 1. But then another progenitor cell will divide, and then crawl up above that first layer. So that these cells are also neurons, but they are the next layer of those neurons in the cerebral cortex. And finally, we've got another cell, the radial glial cell, dividing and migrating, and then it forms another layer even further along above those previous layers. And then finally, we can see that these are the last born neurons, youngest neurons here at the top, and then they sit on top of all of those previous layers of the cortex of the neurons that were born before them. So I think that's really cool, kind of, because it just shows how like these divisions and how those cells work can be steered by where they grow as they divide and as they mature. So anyway, so this is a little bit of an explanation about the nervous system development and how it all works. I hope you guys enjoyed it, and if you've got any further questions, please let me know now. And this is all I've got prepared for you today. So I'm going to grab my phone, and I see a question already here. Let me see. Can you please tell the source? Yes, so the source of those images was the Albert's Molecular Biology of the Cell textbook. I will link it in the story, just so that you guys can see it. It's a textbook which is very popular. It was popular both in my course when I was an undergraduate student, but also in neuroscience courses, genetics courses, cell courses, and cellular biology courses, all kinds of things like this. So I'll link that book so that you guys can check it out. But yeah, it's a really good graph, actually. And yeah, I'm really glad that you guys turned up again to watch my little talk. Next week, I think we might be talking about, I don't know yet, I think we can try to talk about a little bit of an action potential and maybe learn a little bit more about that neural development. I don't know how interesting that would be for people. Radial glia is actually a kind of astrocyte from Zach Dalton. Sorry, I don't know how to read your username. They aren't really an astrocyte. They are an astrocyte precursor. They are precursors to every single cell that happens in the nervous system at that time. So basically, yeah, they will become astrocytes later. They will become oligodendrocytes later. They will become all kinds of other cells, the radial glia. They are a neural precursor cell. Thank you so much. Everybody's saying that it was a good talk. Thank you so much. Anyway, yes, if you've got any further questions, you can message me or reply to the stories. And on the stories now, I will share some more information about the stuff that we talked about. And yeah, it was a pleasure having you guys. And I'm looking forward to our next talk next Saturday. Thank you, Anna. I'm glad you enjoyed my talk. See you guys later.